Hi guys, last section of the chapter, changes in chromosome number and structure. Remember, meiosis almost always proceeds normally. Euploidy is the correct number of chromosomes in a species, and aneuploidy is a change in the chromosome number. A karyotype is a display of chromosomes arranged by size, shape, and banding pattern for observing aneuploidies. Aneuploidy results from non-disjunction, which is the failure of chromosomes to separate. Non-disjunction can occur during meiosis 1 or meiosis 2. It may result in the gain or the loss of chromosomes. Monosomy means only one of a particular type of chromosome, and trisomy refers to 3 of a particular type of chromosome. So here we have a pair of homologous chromosomes. In non-disjunction, the chromosomes do not separate correctly. So here we have 2 chromosomes where we should have 1 and no chromosomes where we should have 1. In normal meiosis, we get 1 in each. If this happens in meiosis 1, when meiosis 2 occurs, we get 2 chromatids in 2 cells and no chromatids in the other ones. If non-disjunction happens in meiosis 2, we get 2 cells that have the normal amount of genetic information, and 1 cell that has double the amount, and 1 cell that has none. And if fertilization occurs, we get a zygote or a fertilized egg that has the incorrect number. So here we would have trisomy and trisomy, monosomy, monosomy. Here we would have 2 normal cells. Here we would have trisomy and monosomy. So here we have labels. All right, so we have a normal diploid plus 1, diploid plus 1, diploid minus 1, and so on. Trisomy occurs when an individual has 3 of a particular chromosome. In humans, 3 autosomal trisomies are viable beyond birth. That means that the baby can make it. The most common autosomal trisomy seen among humans is trisomy 21, which is also called Down syndrome. The chance of a woman giving birth to a child with Down syndrome increases with her age. That's why a lot of older women, whenever they find out that they're pregnant, they have some additional tests to go through to help them see if they have these extra chromosomes. The longer the oocytes are stored, the greater the chances of non-disjunction occurring. When that sperm fertilizes the egg, the greater the chances that that polar body isn't formed correctly. You can recognize Down syndrome by short stature, an eyelid fold, flat face, stubby fingers, and a wide gap between the first and second toes. So here we have a karyotype of trisomy 21. This is the set of chromosome 21, and you'll notice that there is a third one right here. You'll notice that this is not a sex-linked trait. It's not controlled by the X and Y chromosome that can be found here. So the prevalence is the same in males and females. Changes in the sex chromosome can result in different types of disorders. The result of inheriting too many or too few X or Y chromosomes it can happen. It can happen when you get too many X's or too many Y's. Extra copies of sex chromosomes are more easily tolerated than regular autosomal chromosomes, which are normal ones that don't determine sex. Non-disjunction during oogenesis or spermatogenesis, that's when this would happen. Turner syndrome is when a female is born with only 1 X chromosome. Generally, they are short with a broad chest and widely spaced nipples. And they can be of normal intelligence and function normally with hormone therapy. Klinefelter syndrome is when a male has an extra X chromosome, and it results with males with underdeveloped testes and prostate and some overdevelopment of the breast. People with Klinefelter syndrome often have long arms and legs and large hands. They have near normal intelligence unless there are 2 X's or 4 X's or, so on and so on. No matter how many X's, X chromosomes are present, the presence of a Y renders the individual genetically male. So the presence of the Y determines whether biologically they're male or female. Doesn't matter how many X's there are. The deletion of the SRY gene results in an XY female. The lack of testes determining factor, which plays a role in male genital development. So they don't have the right proteins produced. The presence of the SRY determines the maleness. So it's this gene on the Y chromosome that determines male or female. Here we have karyotypes of a Turner syndrome person. You'll notice that there is a missing X chromosome right here. Here we have a Klinefelter syndrome karyotype where there is an extra X chromosome in addition to the normal X and Y. Environmental agents like radiation, chemicals, or viruses can cause chromosomes to break. If broken ends of chromosomes don't rejoin, mutations can happen. Changes in chromosome structure include deletion, where one or both ends of a chromosome breaks off. 2 simultaneous breaks lead to a loss of an internal segment. We're going to see some pictures of that in a little bit. Duplication is when there is a presence of a chromosomal segment more than once in the same chromosome. And translocation is when a segment from one chromosome moves to a non-homologous chromosome. It follows breakage of 2 non-homologous chromosomes and improper reassembly. So 2 chromosomes would have to break and be put back together in the wrong order for that to occur. Inversion occurs as a result of 2 breaks in a chromosome, and the segment is flipped upside down before it is reattached. And the genes occur in reverse order. Okay? They occur in reverse order on that chromosome. Here we have human syndromes. Changes in chromosome structure can cause various syndromes and can be detected in a karyotype or by studying an inheritance pattern in a family. Deletion syndromes include Williams syndrome, which is a loss of the end of chromosome 7, which contains the gene for elastin, which makes certain proteins in your body. And the children resemble pixies. A translocation syndrome is, I'm not quite sure how to say this one, Alagille syndrome is a translocation between chromosome 2 and number 20, which can lead to a congenital heart defect called tetralogy of Fallot. It causes chronic myelogenous leukemia, which is a blood cancer that is caused by a translocation between chromosome 22 and chromosome 9. Here we have deletion, okay, a deletion where A is removed, gene A is removed. Here we have a duplication where D and E are repeated on the chromosome. Here we have an inversion where C and D are flip-flopped. And here we have a translocation where G and H are swapped with Q and R. Here we have a picture of Williams syndrome, okay? And here we have a translocation. Notice the fingernails are not growing in the way that they usually do. And that's it, guys. If you have any questions, let me know.